Great. Thank you so much for having me at this seminar. So, as I was introduced, my name is Nikhil. I'm a PhD candidate currently in the Pritchard Lab. I'm starting my fourth year. Yeah, I'm excited to tell you a little bit about this story about gene dosage response curves in human complex traits. Okay, so our lab is primarily interested in interpreting GWAS results. Um, so these are genome-wide association studies, and they've become a powerful statistical genetics tool to connect variants with phenotype. And the power is that if we control for known confounders that we kind of have a good sense of, we're getting at a causal mechanism. And what that means is if we can understand why the variant has an effect on the phenotype, we're getting some understanding of the causal biology of the trait. So this paradigm is quite powerful, but I don't think I need to convince this audience that GWAS variants are actually extremely complicated to interpret. So here's an example of a very recent GWAS for 5 million individuals. And the trait being studied here is height. So on the x-axis are chromosomal positions. And on the y-axis, this is not a standard Manhattan plot. Each block here represents the number of associations in a 100 kb window. So hopefully you can appreciate that the entire genome basically lights up with signal for height. And so if, let's say, you're a height biologist and I give you this set of variants, it's very hard to figure out where to even get started in understanding the biology of height, right? There's so many genes associated with this trait, so many locations. How do we even understand trait biology? So maybe it's helpful to step back and think about what we mean by understanding the biology of a trait in the first place. So, for example, let's say I'm interested in this trait called blood cystatin C levels. And cystatin C is just a matter of kidney function. So, higher cystatin C is generally worse for you, worse for your kidney. And maybe if I'm a biologist, I want to understand, what is the process that produces cystatin C? What puts it into the blood? What's the degradation process? And there's some other endophenotypes that you might be interested in, like aging, for example. And ultimately, what we really think about is genes, right? We think about gene products and how they interact in how they form these processes that then lead to the trait that we care about. And so, really, the picture we want is at the gene level and not the variant level. And so, uh, one characterization that might be helpful instead of a Manhattan plot, for example, is to think about how the gene directly affects the trait. Right? So imagine I had control over germline gene expression of CST3, which is the major component of cystatin C in blood. So if I could control germline gene expression in humans and I could reduce CST3 levels, I would expect there would be some commensurate reduction in cystatin C levels in blood. On the other hand, if I could increase germline cystatin 3 levels, then maybe I would expect an increase in blood cystatin C levels. Um, and this kind of full spectrum of the response of a trait to titration of germline gene expression levels is what my lab kind of calls the gene dosage response curve. Right? The idea is how does a trait respond to changes in gene dosage of this specific gene. And I want to pause here and just point out, that this continuous relationship already tells us quite a lot of information about the relationship between this gene and trait. So this immediately tells us that this gene is a positive modulator, right? This is, or what you might call an activator of this trait. On the other hand, you could imagine if a curve had a negative relationship like this, it would be a repressor of sorts. We also see kind of the quintessential buffering pattern here. So uh, the you can imagine that blood cystatin C levels maybe can tolerate too much cystatin 3 transcript, but maybe it's very hard to deal with the loss of function, right? So you can see that kind of buffering in this shape. And finally, if you're interested in titrating blood cystatin C levels, let's say you're a drug developer and you're targeting this protein in blood, then this tells you exactly how to modulate the CST3 gene to achieve the effect you want. Right? So there's all this kind of nice information in this curve that you don't get out of a Manhattan plot. Um, so human geneticists don't titrate germline gene expression levels, right? That's not an experiment we actually perform. Instead, what we rely on is natural variation. And there's various types of variants that will give us different effects that we care about. So for example, deletions and loss of function variants in genes will reduce expression for us, and then we can observe their effects on traits. Duplications, on the other hand, increase gene expression levels for us. And finally, we also have a class of variants called EQTL. These tend to be non-coding, and they titrate gene dosage around the population mean. And so if there's one thing I want you to take away from this talk, it's this kind of picture here. So instead of a Manhattan plot, the idea is we have this dosage response curve, and every variant we really care about falls along this dosage response curve for some gene somewhere, right? So these loss of function variants that I told you about reduce dosage, right? Duplications increase dosage, and EQTLs kind of titrate around the population mean. So the information on the x-axis comes from either knowing what the variant does already or measuring it through an assay, like an EQTL expression assay. On the other hand, the information on the y-axis is coming from some type of GWAS analysis. So understanding how much a variant affects a trait comes from performing some type of genome-wide association study in a big cohort. Okay, so from here, I'll move on to my actual project here. So my lab likes to use the UK Biobank. That's kind of our cohort of choice. And so we ran what are called loss of function and duplication burden tests. And these rely on using the whole exome sequencing and whole genome sequencing data in the UK Biobank. So if you're not familiar with this type of genetic variation, loss of function variants are coding variants. So they're present in the gene coding sequence, and they result in nonsense-mediated decay. And so we expect these to reduce dosage on average. On the other hand, duplications are, in this study, are whole gene duplications with some flanking regions. So we expect these to increase gene dosage on average. And in the UK Biobank, around 17,000 genes have loss of function variants. And of those, around 9,000 genes have duplications. So we're dealing with a set of 9,000 genes, if that makes sense. So how do you perform a burden test? Basically, you separate individuals who have a loss of function in a gene versus those that don't. So for example, these are individuals that have one copy deleted versus not. And then I measure the average phenotype value in these two sets of individuals. And the regression line tells us what the effect is of deleting that gene. So similarly, we can do the same for duplications. I can separate individuals that have duplications in a gene versus those that don't. And then the effect estimate from regression tells me what is the effect of duplicating that gene on the trait. For those that are curious, you might know that duplications tend to carry more than one gene, and there's a bunch of statistical stuff we do to account for that. So if you're curious about that, you can ask me later. But here's kind of a quick view of what the data looks like, right? So here's actual real data now, and I'll just focus on this curve right here for SHOX. So the dashed line is hypothetical, just to be clear, but these 2 points are actual estimates from our study. So here's the effect of deleting the SHOX gene on height. So on average, deleting this gene seems to reduce height by almost 1 standard deviation. And duplicating also has a significant effect, but it's much tinier. And so we can imagine that this gene is a positive modulator of height. And here's a classic example of deleting LDLR, which is the LDL receptor. So deleting LDLR, we do expect it to increase LDL, and duplicating it, we do expect it to decrease LDL. This is a very well-characterized gene. So both of these are well-characterized genes. So this all sort of makes sense. But burden tests are actually extremely noisy. So I ran these tests for around 160 traits, complex traits in the UK Biobank, and across all the 9,000 genes that I mentioned. And here on this slide are all the top hits you can find genome-wide. So it's a very noisy set. I showed you two of these. I showed you SHOX, the gene on the last slide. So here's what happens when you delete this gene, and here's what happens when you duplicate that gene. You can see that for most gene trait pairs that we find are significant, deleting and duplicating genes have opposite effects. So this in our lab we call this a monotone relationship because if you look at the curve on the last slide, the curve is increasing, which is called monotone. These are called monotone curves. But you'll also see there's two examples here where deleting and duplicating the gene has the same effect on the trait. It's in the same direction. And this is called a non-monotone relationship. Um, so these are interesting patterns, I think. But because the tests are so noisy, we have to start summarizing the information somehow across genes. And so one summary is to take the mean effect of all genes for a trait. So now let me walk you through this plot because this one is quite important. So here, each point represents one trait. So for example, cystatin C levels or BMI or height, for example. And on the x-axis, I'm showing you what is the average effect of deleting a gene. And on the y-axis, I'm showing you what is the average effect of duplicating a gene. And you'll notice something quite surprising, actually, which is that deleting and duplicating genes on average tends to have the same direction of effect on a trait. So for example, for height, deleting a gene tends to reduce height on average, but duplicating a random gene also reduces height on average. And this to us initially was kind of surprising because just on the last slide, I showed you that most genes have opposite effects. And so somehow after aggregating all these opposite effects, we have come to a situation where the average effect is in the same direction. Okay, so we're going to walk through this using kind of the curves that I introduced at the start. So again, this is the graph from the last slide. And we're specifically going to focus on this point FVC, which stands for forced vital capacity. And one point I want to make is that this point represents the average gene dosage response curve. So imagine this point right here. It has some x-axis value and some y-axis value. You can actually plot those out as an average curve like this. And the idea is that all of these curves in the background are gene level curves. And once you average these gene level curves, you get this average gene dosage response curve. And that's what I'm plotting here on this plot. So the observation we've made is that all of these average curves tend to be U-shaped or non-monotone like this. And we propose that there's basically two models that can explain why the average is U-shaped. One is that most of our genes are monotone, which means deletions and duplications have opposite effects, but they're all depressed at the top like this. And so when you take their average, they end up making this U-shaped curve. The other explanation is that there are some genes which have non-monotone relationships, but there's more of them in one direction than the other. So the rest of the study is really thinking about which one of these really explains the data. Okay. So one very simple idea we had was estimating the correlation between deleting and duplicating genes. So why does this work? Here's a quick schematic to explain how this works. Imagine I make a plot of the deletion effect versus the duplication effect of a gene. If the gene is monotone increasing like this, then it falls in this quadrant right here, right? Because deleting it reduces the trait, but duplicating it increases the trait. And so you can take my word for it that these red quadrants have monotone gene dosage response curves. On the other hand, these two quadrants will have non-monotone gene dosage response curves. And so if I estimate the correlation of the genes on this plot, if the correlation is negative, most of the GDRs, a lot of the signal is monotone, basically. On the other hand, if this is positively correlated, a lot of the signal is non-monotone. And so we just take the negative correlation, basically, and that's our monotonicity estimate. There's a bunch of statistical machinery that goes into this that I'm sweeping under the rug. But if you're curious, you can reference our preprint for how this works. But here's basically the results from this analysis. So again, each bar here represents the estimate from one trait. Here's our estimate of monotonicity. And the lines in dark, these are 95% confidence intervals. And the lines in dark black are ones that are nominally significant. And you can hopefully appreciate that all of the signal, basically, especially the significant signal, is on the right, which means that for most traits, genes that affect those traits tend to be monotone, right? So the deleting a gene tends to have the opposite effect of duplicating a gene. So this is where biology makes sense, right? Like this is what we expect from most genes, and this is indeed what we see across traits. Okay, so let me circle back to this model. If we really look at these curves that I've stylized in the background here, the red and the gray curves, you'll notice that uh, they're all depressed in the same direction, right? So the way to think about that is if you're trying to, what I'm plotting here is forced vital capacity. If I task you with finding a gene that decreases forced vital capacity, it's easy pickings, right? Most of these genes you can titrate in some way to reduce forced vital capacity quite a lot. But if I ask you to increase forced vital capacity, you're going to have a pretty hard time finding a gene that does that sufficiently for you. So we call this phenomenon trait buffering because it's buffering against one trait direction. In this case, we're buffering against increasing forced vital capacity. And so to measure this property, uh, what you'll notice here, here I've stylized these curves that are all depressed in one direction. And this is our prediction for height, right? For height, everything is going to be depressed against increasing height. And what that ends up looking like on this plot is that all of the genes are going to fall below this diagonal line right here. This is our prediction. We actually wanted to look at gene level estimates, which is what I'm going to show you in just a second. So to do this, we used an empirical Bayes approach. Again, the details I'm sweeping under the rug, but feel free to ask me later or check our preprint for how we actually do this. But here's now data for height. Here, each point represents an actual gene. So I've actually made this plot that I've been talking about right here for real data. Here's the diagonal line I was talking about. And these big circles represent genes that we're somewhat confident in. And hopefully you can appreciate that there's very few genes above the diagonal line, but a lot of genes below this diagonal line. Right? So this is kind of recapitulating this picture of these gene dosage response curves for height. Okay, so that's one trait, but we can analyze this across traits, and then we can also decompose this signal into monotone and non-monotone bins. And this figure is quite busy. I apologize, but actually, I just want to draw your attention to this graph right here for height. So as we go along this graph, we're getting increasingly confident in the sign of the gene. And so you can just think of this as increasingly stringent significance cutoffs. And here I'm decomposing the signal into monotone and non-monotone bins. And hopefully what you can appreciate is that both non-monotone and monotone gene dosage response curves seem to contribute to this trait buffering that we observe in the data. So to tie it all back together, you know, this idea that the average GDRC is U-shaped can be explained by both of these curves, although most of the signal is present in these monotone gene dosage response curves. Okay, so to conclude, hopefully I've convinced you that maybe instead of a Manhattan plot, we should be thinking about these gene dosage response curves because ultimately we care about the effect of genes on trait. And so it might be helpful for us to put these GWAS variants onto these curves so that we can understand how genes are affecting traits instead of just variants. And there's many different ways to do this. So for one of my future kind of projects I'm thinking about is we haven't used eQTL at all yet, but there's a whole class of methods that deals with this exact question. You might have heard of transcriptome-wide association studies or Mendelian randomization. Ultimately, those methods are actually estimating the GDRC at the origin. And so we can think about using those methods in this context as well. The other things I showed you are that for most trait-relevant genes, they are monotone, which means they are more like this red curve than this gray curve. So that's good. That's kind of, I think, what we expected from the start. And then I also showed you that there's this kind of buffering that's happening, where for some complex traits, it's easier to decrease their value, but harder to increase, or vice versa. It depends on the trait. But there's this buffering going on against one direction. Uh, and there's a lot of evolutionary stuff that I've kind of ignored as well for this presentation. But if you're interested in why we might see these kinds of architectures in real traits, we have some discussion of this in the preprint. So feel free to reference it there. Okay, so I just want to thank my lab. Here's our current most recent picture. These are my current co-authors on this project. I would especially like to thank Jonathan, of course, and Jeff, who was my direct mentor for this project, and also my thesis committee. And yeah, again, just a pitch for the preprint if you're interested. So thank you. I'll take any questions.
Thank you. Is there any questions from the audience? You could just type in the Q&A or in the chat. I see it's none, so I asked two questions. Um, so first, I think I missed the point because at some point you should apply the averaging all the burden tax effect
Mhm,
across all the genes for both loss of function duplication. And you show that those effects are in the same direction. But
mhm,
also remember, you show a lot of plots that if you look at one gene, we do see the two effects are opposite.
mhm.
So I'm just a little bit confused. What is like your center conclusion on
Yeah.
this?
That's a good question. So yeah, that comes back to this kind of plot right here, right? The observation that averaging across all genes has the same direction of effect is represented by this black curve right here. And when we average across a bunch of the what I'm showing you here are two models that might explain this observation. So let's just look at this model, which kind of matches our biological intuition. So each red curve here represents one gene. And you can see that each of these genes has opposite effects, right? Like duplications and deletions have opposite effects. But I've drawn them in such a way that if you average all the points here, you'll notice that the average is below zero. And then if I average all of the points on this area, they'll also fall below zero, right? So we're arguing that the architecture of these curves might look something like this, so that at the gene level, they're truly opposite. But once you average them, they end up going in the same direction.
Oh, I see. And
Yeah.
for the non-monotone model, it's one specific gene is non-monotone.
Yeah, and those have to be in one direction more than the other, right? So here you'll see all of these curves are non-monotone, but more of them have to be in one direction than the other. Yeah, yeah, yeah.
Okay, great. Very
Cool.
interesting.
Yeah. No, thank you for asking. Yeah, that's probably the central point in some sense. Yeah.
Yeah, and okay, so this is one thing, and the other okay, there's no problem in the chat. Okay, so the other things that you mentioned, another statistics, which is the trait buffering.
Hmm.
So is that trait buffering on a single gene or on the average of all the gene?
Oh, that's also a good point. Yeah, yeah. So the trait buffering is averaging across all genes as well. So we are very, very underpowered to call anything for a specific gene. So yeah, all of these results that I'm showing you here are averaging across genes. Um, yeah, unfortunately, uh, we're talking about variants that are super rare, right? Essentially. So uh, yeah, like all of these estimates are from 9,000 genes, basically.